Good afternoon, and thanks so much for joining us today as we kick off our webinar series on improving the health span of older adults through nutrition. My name is Wendelin Jones, and I am the Executive Director here at IAFNS. We're so thrilled to be doing this event with Tufts and the John Mayer USDA Human Nutrition Research Center on Aging. Let me first start with a brief reminder on who IAFNS is. IAFNS stands for the Institute for the Advancement of Food and Nutrition Sciences. We mobilize government, industry, and academia to drive, fund, and lead actionable science to advance public health. We catalyze the creation of scientific knowledge and convene experts to drive positive change. Our collaborative and inclusive structure, bringing together government, industry, and academia, empowers all of our members to bring forward the diverse perspectives that make up our food and beverage ecosystem. As an organization, we are grounded in our 4 core values: scientific integrity, transparency, collaboration, and public benefit. Using those core values as the guide, we're able to include the diverse perspectives that is crucial to the development of credible science. Our focus is on connecting scientists, collaborating with diverse multi-sector stakeholders, catalyzing science to deliver science that matters. With our platform today, which is the GoToWebinar platform, wanted to take a brief minute and familiarize everybody with the controls in case they weren't aware. In the attendee box on the upper side, you have a red arrow. You can collapse that attendee box if you would like to see more of the screen. Additionally, and this is actually very important, within the meeting platform, there is a questions box. We encourage you to type in questions into the questions box. At the end of today's presenters, after they're done, we'll be reading all of those questions and allowing for a dialogue that way. We've got almost 600 folks registered for this event, so you can understand why we have to go with typing in questions as opposed to unmuting and letting folks answer. But we're thrilled that we have so many registrants coming in today for the meeting, because we've got 3 very, very exciting presenters ready to talk about their work. We're going to hear from Dr. Sarah Booth, Dr. Christopher Wiley, and Samin Madani on their work. So without further ado, I'm going to transfer presenting rights to Dr. Sarah Booth. And while I'm doing that, here we go. I'll quickly note that Dr. Sarah Booth is the Center Director at the John Mayer USDA Human Nutrition Research Center on Aging at Tufts University, a position she's held since 2018. She's one of the world's leading experts on vitamin K and is currently the principal investigator on a National Institute of Aging grant that's examining the association between vitamin D and vitamin K in Alzheimer's and other dementia in older adults. She is a leader in the nutrition community and currently serves as treasurer of the American Society of Nutrition. She's also deputy editor of an open access journal, Current Developments in Nutrition. So with that, Dr. Booth, the floor is yours.
Thank you very much. Thank you, Wendelin. And I just want to tell everyone that the Jean Mayer USDA Human Nutrition Research Center on Aging at Tufts University is so excited about partnering with IAFNS on this webinar series. We share a common passion with IAFNS for bringing together many sectors to explore common interests, such as the health span of older adults in pursuit of new scientific discoveries. So for the rest of my talk, I am going, to just turn off my camera so you can focus on my slides. Here are my disclosures. So let's get to the purpose of our, there we go. Of my talk. So why are we focused on the topic of improving older adults? That question will be answered in each of the 4 webinars in this series. But the uber answer is in the demographics. Last year, we passed a milestone. Globally, there are now more people over the age of 65 than under the age of 5. Very soon, in this decade, in fact, more than 1 in 5 people in the United States will be over the age of 65. Think about it. 1 in 5 people will be over the age of 65. Now, when my grandmother, who used to live in Western Australia, reached 101 in January of the year 2000, it was such a novelty. She was featured on the front page of the local newspaper to welcome in the new millennium. Right now, there are currently just under 100,000 centenarians in the United States. By the year 2060, which is less than 40 years from now, the numbers of centenarians will have grown to 500,000, and that is in the US alone. In other words, older adults are the fastest growing demographic in our country, and indeed globally. We need to accelerate our research to meet the needs of the population that is living longer, but not necessarily in good health. Our aging population is living with multiple chronic diseases. That reality is the theme that will run through this webinar series. And we will focus on how to reduce this huge public health threat. Right now, less than 20% of all adults over the age of 65 have no chronic disease, just 20%. The US simply cannot sustain spending upwards of 75% of its healthcare dollars on treating chronic diseases. Indeed, I am hopeful the COVID-19 pandemic will result in a greater appreciation of preventive and public health approaches to addressing the health and well-being of our older population. So let's dive into the complexities of the biology of aging. We do tend to focus on lifespan, particularly in the popular press and also in our research. But should the question be, how long do we live? Aging is multifactorial. Genetics and the environment all contribute to our rate of aging. Most of this webinar series, though, will be focused on how nutrition and lifestyle influences healthspan. Healthspan, not lifespan. Healthspan, in contrast to lifespan, is generally defined as the period of a person's life when they are in good health. For most, that is when they are free of disability. Indeed, when you ask a centenarian what is most important to them, they will not say they want to live longer, no matter what. They will tell you that they only want to live as long as they have quality of life. Throughout this series, you are going to hear how age is clearly a risk factor for many chronic diseases. Simply put, most of us develop chronic disease when we get older. However, there are ways to mitigate that risk of chronic disease. So here in this figure, you see how function improves and then declines with age. So let's use the example of bone. We reach peak height and peak bone mass in the first 3 decades of life, and then bone declines thereafter. The challenge is when the function declines so much that one passes from limitations, so in the example of bone, that would be low bone mass, to frank disability, such as osteoporosis and hip fracture. Nutrition and lifestyle can delay the transition of declining function. One way can be to delay the trajectory or the slope of the decline such that the years spent in disability are markedly truncated. Alternatively, one can adopt strategies early in life to build more reserve. Hence, the trajectory of decline is the same, but there is simply more to lose. Hence, there are fewer years spent in disability. So how do we achieve these models of extending health span at the HNRCA? We study healthy aging and focus on the influence that food, nutrition, and physical activity have on slowing the progression of disease and the vital role they play in allowing people to live long, active, and independent lives, free of disability. In this virtual world, I want to visually place you at our center. This is a picture of our building located in the Health Sciences campus of Tufts University in downtown Boston. Our center operates through a cooperative agreement between Tufts and the USDA's Agricultural Research Service. There are 3 driving forces, 3 dimensions to everything we do at the HNRCA. The first dimension, or D, is discovery. This drives our research agenda, and this is indeed what we will focus on in the next 4 webinars. Our center focuses on many key issues involving aging, and as you will learn, we work very collaboratively. It's critical that we take a systems approach to studying healthy aging, and that is why we are moving away from exclusively studying single nutrients or individual organ systems. The field of geroscience, which is the study of aging, has revealed in recent years that the forces that influence, for example, aging of the bone, can also influence, for example, the aging of the muscle. Same mechanisms, different chronic diseases. And remember that the majority of older adults have more than one chronic disease. Therefore, by targeting the root causes, multiple chronic diseases can be addressed with a single strategy. The second D, or dimension, of the HNRCA is develop. We are committed to developing the next generation of investigators, and our work is enriched by the Tufts graduate students we train in our labs. We also have a vibrant community of postdoctoral scholars that are an essential part of our research enterprise. Furthermore, the HNRCA is investing in early stage investigators. And indeed, today you will be hearing from our newest addition to our scientist community. In future webinars, you will also be hearing from some of our trainees. The third dimension to the HNRCA is deliver. We are committed to bringing our research into the many communities that may benefit. This webinar is just one example. Pre-pandemic, as depicted in this slide, we actively engaged in community outreach to disseminate our research findings. Over the next several weeks, you will meet 11 members of our research team. Of course, each investigator and every important research finding is supported by many more people than just our speakers. So I wanted to share this pre-pandemic photo of all the folks who contribute to the success of our center. I do hope you find this webinar series informative, and perhaps it will inspire new research collaborations within and among industry, government, and academia. Thank you very much, and I hand it over to my host, Wendelin.
Gwendolyn, you're muted, just to let you know.
I was saying all brilliant things. Just know that. So I was saying thank you, Sarah, for that wonderful introduction to this webinar series on making sure that research is extended broadly into communities. Also, thank you for sharing the good work that is done at HNRCA. So with that, and as we're seeing these slides come up, I'd like to briefly introduce Dr. Chris Wiley. He joined HNRCA in October after finishing his postdoctoral work at the Buck Institute. Accepting the HNRCA position required that Chris relocate from California to Boston in the middle of the pandemic and just as winter was starting. This clearly reflects his dedication to science and doing what's necessary to pursue his areas of interest. His research is focused on aging in the context of cellular metabolism, particularly as it pertains to a process known as cellular senescence. So with that, Chris, please.
There we go. Thank you so much, Wendelin. I appreciate that. So as Wendelin mentioned, I'm the newest scientist here at the HNRCA, and I'm more of a basic biologist, which means that I study the basic underlying processes that drive aging, particularly one known as cellular senescence. Okay, great. So just to get my disclosures out of the way as well, I have a number of US patents from my time at the Buck Institute. Even though I'm a basic biologist, it still turns out that there's a lot of translation that you can get from basic biology, and that is reflected in these patents, which I think hopefully will lead to translational interventions in the future. So it probably doesn't surprise anyone that age is the single biggest risk factor for a whole bunch of degenerative diseases. Whether we're talking about Alzheimer's and Parkinson's in the brain, or graying and hair loss in the skin, or macular degeneration in the eye, this and a large number of different processes all have at their core age as their #1 risk factor. And a classic example of this comes from cancer. So most people know that if you smoke, you're likely to develop cancer, and there are a whole bunch of, oh, excuse me, I'm having trouble finding my cursor. Well, I'm gonna do this without a cursor, you'll have to excuse me. So most of us know that smoking can contribute to developing cancer, but if you look at all of the major risk factors for developing cancer, they are all overwhelmed by that red bar there, which is just being over the age of 55. That age is your #1 risk factor for developing cancer, but it's traditionally rarely discussed. And the reason for that is that it was traditionally believed that there was nothing you could do about aging, so why discuss it as a risk factor? But this is really a changing perception now. And if you go to the National Cancer Institute's website, and when they talk about risk factors, age is the #1 risk factor that they bring up. So we know that these degenerative diseases rise exponentially with age, and we don't think this is a coincidence. There have to be one or more basic aging processes that drive aging, and most or all of these age-related diseases. And as Sarah brought up, this led to this idea of a geroscience hypothesis, this idea that if we can target those basic processes that drive aging, we can intervene in age-related disease and change that slope of that curve towards developing fewer age-related diseases and living not only longer, but healthier. So we've identified quite a few basic processes that drive aging. These include a large number of factors, but I focus right here in this one right here, which is cellular senescence. But I don't want you to think that these processes are acting independently of each other, because all of these processes here can drive cellular senescence. Whether we're talking about things like genomic instability or the ever popular telomere attrition or mitochondrial dysfunction, which I study, all of these processes drive cellular senescence. Cellular senescence in turn can drive both stem cell exhaustion and altered intercellular communication. And because of that, I like to think of senescence as a nice point for intervention because it's at the nexus of so many basic aging processes. So just to summarize what I'm going to tell you about today, first I'm just going to introduce you to the concept of cellular senescence, and then I'm going to talk about the relationship between senescent cells and the diseases we associate with aging. And finally, I'm going to give you a little bit of hope in showing that there are interventions that we can use to target cellular senescence. So what is cellular senescence? Well, as I described, it's a basic aging process, and the key point is it's a stress response. Cells become senescent in response to a certain number of stressors. These include things like genomic or epigenomic damage, so things that harm DNA or the control of DNA, mutations that activate oncogenes, which are genes that promote cancer, can also drive senescence, as do metabolic imbalances and organelle stress, including mitochondrial stress. So the key features of senescence include an irreversible growth arrest. So a senescent cell will never divide again. It stops dividing, and it adopts this arrest, this dividing arrest. That means it never will divide. So that you could imagine if that's a cell that could become cancerous, it's really good that it stops because that's not going to form a tumor. But on the other hand, if that cell is a stem cell, it also might be bad because that cell is no longer going to populate a tissue. It also has this multifaceted secretory phenotype, or the senescence-associated secretory phenotype, or SASP for short. And this is a collection of pro-inflammatory cytokines, chemokines, growth factors, proteases, lipids, and other molecules that are secreted by senescent cells that can influence the cells around them. One key note for the rest of my talk is that senescent cells can be stained and identified by the activity of what we call senescence-associated beta-galactosidase. And what that allows us to do is give the cells a compound called XGAL that is cleaved by beta-galactosidase, and when we do that, the cells turn blue. So throughout the rest of my talk, I just want you to associate blue with senescence, okay? So how do we think senescent cells might drive aging? Well, there's that growth arrest. That's a cell autonomous process. That means that it only influences that individual cell. But if that cell happens to be a progenitor cell or a stem cell, that cell is no longer going to contribute in any meaningful way to a tissue. So if you're thinking about tissue regeneration, that's going to impair tissue regeneration. However, we think the SASP, which is cell non-autonomous, in other words, it influences other cells, is probably a bigger driver of age-related pathology. And that's because those secreted factors include lots of cytokines and chemokines that can drive sterile inflammation. It can disrupt the extracellular matrix, and it can result in other perturbed signaling events that together we think can cause pathology. So we've identified quite a few different factors that are increased with the SASP. These include all these things you see in yellow here on this slide. The images to the left indicate factors that are upregulated by yellow with senescence. Those are all proteins, and most of those are pro-inflammatory cytokines. In other words, the SASP drives inflammation, which is a sterile inflammation. It doesn't have any kind of virus or bacterium or anything else associated with it. And this occurs more with age. So what we call this is inflammaging in short. We also see on the right, those yellow upregulated factors are lipids that are being upregulated with senescence that are also tend to be pro-inflammatory. So we know that senescent cells increase with age in most tissues, and we've seen this in almost every vertebrate. We've seen senescence increasing in fish, in frogs, in mice, and in humans. Pretty much across the board, in every vertebrate we've looked at, and in virtually every tissue we've looked at, we found senescent cells. So we see senescent cells in the bone in the case of osteoporosis. We see senescent cells in the brain in the case of Alzheimer's. They show up in many different places. And you can see there those blue cells in the middle right panel. I apologize that I don't have my cursor here. But you can see those blue cells showing up there in the old dermis in this individual. So senescent cells are showing up at the right time, and they're showing up at the right place to cause aging and age-related disease. So the question for a long time was, okay, do they? Because these are basically just suspects. They're showing up and they have all the method and motive to cause disease, but do they actually drive disease? And I think we know the answer to this now. And this is work that came out of the Mayo Clinic, where they engineered a transgenic mouse where they expressed a transgene from the P16 promoter. So that all you need to know is P16 comes up with senescence. And so it's a subset of all senescent cells, but a really important subset of all senescent cells, because when they expressed this transgene, which allowed them to selectively kill senescent cells with a drug, abbreviated here as AP, they saw something pretty interesting. So if you look at the image on the left, you'll see that we get on the y-axis is survival, on the x-axis is time. So when you see a shift to the right, that indicates lifespan extension. And I'm going to show a lot of these things, so I just want you to keep remembering that the shift to the right equals lifespan extension. When we see that, when these mice were given this AP and had their senescent cells eliminated, they lived longer. But it wasn't just that they lived longer. If you look at the mice, you can see an obvious indicator. The mice that do not have AP look old. They lose their hair, they're hunched over, they are old mice. Whereas the mouse on the right, which has had its senescent cells removed, looks younger, behaves younger. It looks like a happier, healthier mouse than the one on the left. So we know at this point that senescent cells limit lifespan and can cause age-related pathology. But what about other diseases of aging, like I talked about in my earlier slides? Well, we know that diseases that mimic aging, such as Hutchinson-Gilford progeria, which is a horrible disorder in which young children develop age-related pathologies, as you can see here on the left. This girl doesn't have any hair anymore, and these kids tend to die of age-related conditions before the age of 20. It's really a horrible disorder. Well, you can see that if we take cells from these kids and we compare them to cells from healthy individuals, they tend to have more senescent cells. So you see fewer cells there, and they tend to be staining blue again for that senescence-associated beta-galactosidase I told you about. And an example from my own work, something that's well known in the HIV field, of course, is that HIV shortens the lifespan of infected individuals. So that blue line there is the uninfected population. The red line is people who are infected with HIV but do not receive any sort of therapy. And of course, if you don't receive any sort of therapy and you're infected with HIV, you develop AIDS, and at some point you'll pass. However, in the advent of antiretroviral drugs, people no longer get AIDS and they live longer. But you'll notice that that lifespan curve in yellow is not the same as the uninfected population in blue, indicating that there's still something that's limiting their lifespan. And when these people show up in the clinic, they show up with a lot of disorders that collectively resemble premature aging. So one of the things I decided to test was this idea that maybe these HIV therapeutics might drive senescence and perhaps some premature aging. And it did work quite well. So TDF and FTC here is a combination of two drugs that are known as tenofovir and emtricitabine. And they are marketed to people who are not currently infected with HIV, but might become infected as something called pre-exposure prophylaxis. Furthermore, these drugs are also used for suppressive therapy in combination with other drugs for people who do have HIV. So it occurs in both cases. And what you can see is when we give these drugs to these mice in their drinking water, they show signs of premature aging, including spinal curvature, loss of hair, and even some hair graying. And when we give these drugs to human cells, we also see that they become prematurely senescent, which is those blue images of cells down below. So the bad news is senescent cells cause or contribute to a large number of diseases, including Alzheimer's and Parkinson's disease in the brain, atherosclerosis and cardiovascular dysfunction in our cardiovascular system, cancer metastasis and recurrence. Furthermore, they mediate the negative effects of chemotherapy, including cardiotoxicity, blood clots, and fatigue. In the eye, senescent cells drive cataracts and retinopathy. In association with diabetes, senescent cells can drive diabetes, but they can also drive diabetic complications as well as other forms of metabolic syndrome. Senescent cells can drive alterations in the immune system in the form of myeloid to lymphoid skewing. They can drive pulmonary fibrosis, osteoarthritis, osteoporosis, and sarcopenia and frailty. That's a lot of diseases, and I just want to point out that the ones I have in red there are just ones I personally worked on. But I also want to point out that there are other people working on these at the HNRCA as well, and you'll be hearing more about them in upcoming talks. Finally, I just want to point out that it's not all bad. Senescent cells do actually have good properties. So when you injure yourself, if you get a cut, you will have senescent cells appearing at the site of that cut, and they help in wound closure and wound healing. They're also involved in tissue regeneration. For example, if you lift weights and you kind of tear up your muscles a little bit, senescent cells help you build new muscle. They also appear during human embryogenesis and during parturition. In other words, they are essential for live birth. So you don't want to just think that senescence isn't all bad, but it certainly can drive bad diseases. But there's also some good news here. We can actually intervene in many forms of senescence-driven pathology. And there are three ways we might consider doing this. The first one is the easiest, which is to prevent senescent cells from occurring in the first place. The second one would be to target the SASP. So you might still have senescent cells, but they wouldn't be able to do as much damage. And the third one would be to kill the senescent cells with compounds that we call senolytics. So the easiest way to avoid senescence, of course, would be to prevent the accumulation of senescent cells in the first place. And we know that we can do this. One example of this comes from caloric restriction. So caloric restriction is known to extend lifespan in a large number of different species, everything from yeast and worms and flies to mice and even non-human primates can experience caloric restriction when we limit the number of calories they consume. And you can see this here for actual mouse lemurs on the upper right there, the red pane indicating extension of lifespan relative to control animals in blue. We also can see that senescent cells are influenced by caloric restriction as evidenced here in the kidney below. So you have sections of the kidney. The first one is control, those are normal aged kidneys. Second one is caloric restriction or CR, where you see less blue staining, again, less senescence. And then furthermore, HC or high calorie diets drive more blue, therefore more senescence. But of course, dietary intervention is difficult. We've known for decades that things like exercise and diet can be good for us, and yet we're dealing with an obesity epidemic here in the United States. So it might not be universally useful as it requires changes in human behavior. So one alternative approach might be to block the SASP and prevent some of those negative effects we see with senescence. And one of the first drugs that we've found that does this is called Metformin. So Metformin is commonly prescribed for people who are either diabetic or pre-diabetic because it can be used to lower blood glucose. It also happens to be the first drug that was used specifically in a clinical trial that targeted aging. It's otherwise known as the Targeting Aging with Metformin trial, or TAME trial, T-A-M-E. So if you're interested about that trial, it's very interesting. Look it up. It's very easy to find literature on that. So we know that metformin extends lifespan in mice. That's that middle panel there. Again, the solid line indicating metformin, the dotted line indicating a standard diet. And we finally know from cultured cells that if we give them metformin, it prevents much of the pro-inflammatory features of the SASP, as indicated by the shift from dark black to red, indicating lower levels of all those pro-inflammatory cytokines you see at the bottom of that heat map. Rapamycin is another drug that has very similar effects. Rapamycin is a drug that was found as a compound that is made by a soil bacterium on Easter Island. Easter Island is also known as Rapa Nui, so that's where rapamycin gets its name from. And it's often given right now to suppress the immune system during people who have either organ transplants or other forms of implanted devices to prevent immune rejection. And so what I want you to look at on the left-hand side is you have normal cells on the far left in blue, all that blue stain, or all that blue indicator on the heat map. The far right of that one is yellow. Those are all elevated factors, in other words, SASP factors. In the middle, you see rapamycin, where it's lowered many of those factors down. And we know that rapamycin can extend lifespan in mice. That's the two images to the right, one of which is from males at the top and on females at the bottom. So it works regardless of gender in mice. So we know rapamycin is very good for that too. Finally, for my own work, there's a endogenous metabolite known as alpha-ketoglutarate. You probably learned about it at some point in high school biology because it's a part of the Krebs cycle. But if you're like me, you probably forgot about it after that. But one of the more interesting things about it is even though it's made at trace levels in our bodies all the time, we can give it to animals just in their drinking water in high amounts. And what you can see in the upper heat map there is that you have 18-month-old or middle-aged mice have a certain pro-inflammatory profile that's relatively low. But at 18 months of age or strongly aged mice, you see that red indicating increases in all those pro-inflammatory cytokines, which come down if the mice are given alpha-ketoglutarate at the bottom. And on top of that, when we look at the survival of those mice, you can see that they have an extended lifespan, as indicated by that shift from the black, which is the control, to the red, which is alpha-ketoglutarate. I also find that there are, so the one other thing to consider though is all three of the interventions I talked about here, you have to consistently take. You'd have to take these at least daily in order to see these effects, because that's what we did with the mice. So that might not be ideal from the standpoint of that's just constantly dosing yourself. Maybe instead what we want to do is just get rid of the senescent cells altogether. And that's where senolytics come in. And these are compounds that kill senescent cells. And one of the first ones that was discovered is called physdin. It's a flavonoid found in strawberries. You could theoretically eat enough strawberries to get this effect, but don't do it. You won't like it. It's about 4 pounds of strawberries in one sitting. Interestingly, if we give it to mice, we see that here again, these mice are in, the white mice are in blue, blue indicating more senescence. You see that the control mice have lots of senescence. The mice that are given the physdin have less. And then if they start mice on a diet of physdin at middle age and take them out and let them age, they live longer than the control mice, showing that physdin does in fact extend lifespan as well. And work coming here from the HNRCA is in my lab, we're working on a new senolytic compound, which is actually an endogenous lipid, fatty acid that is found in the body and can be elevated by consuming things like evening primrose oil. And we find that if we give this lipid, which is dihomogamma-linolenic acid, or DGLA for short, to these mice, we can show that we have both a lowering of it. This is the same aged mouse on the before and after. So before being treated and after being treated, you can see the lowering of the number of senescent cells. And then if we take the tissues out of the mice and stain them for beta-galactosidase, again, we see with DGLA, there's almost no senescent cells in the fat, whereas the aged mouse that didn't get DGLA has plenty of beta-galactosidase in that blue staining. And we see very similar results in the liver. So just to summarize what I've told you today, cellular senescence is the basic aging process, and it can drive sterile inflammation and other forms of chronic degenerative diseases. We can target senescent cells in multiple ways to delay aging, which is why I think that it's interesting to study basic biology, because sometimes basic biology still teaches us things that are useful for interfering in the processes that we study. And those include preventing senescence by using dietary interventions, blocking the SASP to prevent the development of pathology, and then finally using senolytics to eliminate those senescent cells altogether. So there's still a lot of work to be done. We don't know every disease that's being driven by senescence, and it just seems like every month there's a new disease that has been discovered to be driven by senescence. Just this past month, there was a paper on non-alcoholic fatty liver disease being driven by senescence. And of course, from my own viewpoint, I want to know whether senescent cells are actually driving those HIV-associated premature aging phenotypes. We want to know how many different kinds of senescent cells there are. Is senescence like cancer, where there's a lot of different types of cancer, and therefore you'll need to use a lot of different types of chemotherapeutics? Or are they very similar to each other, and therefore would senolytics be very broadly active? And you could imagine situations where you would want them to be very specific. For example, if you're injured but also have to recover from a chronic disease, it might be nice to be able to just target the chronic disease while at the same time being able to heal your wounds properly. And for my own work, of course, we want to know how DGLA kills senescent cells. We haven't really worked that out yet. Finally, we don't really know why caloric restriction prevents senescence. We only know that it does. And it's just one dietary intervention. Are there other dietary interventions we can use to target senescence the same way? With that, I'd like to thank you for your attention, and I'll hand things back to our host.
Fantastic. Thanks, Chris. That was really fascinating. I think we've got all of us thinking about senolytics and what this all might possibly mean for enhancing aging and increasing the health span of folks as they age. So with that, it is my privilege to introduce Dr. Samin Madani. She is a senior scientist and leads the nutritional immunology team at the HNRCA. She was also the center director of HNRCA for 7 years before she assumed the position of vice provost for research at Tufts University. She has since stepped down from that vice provost role in 2019 and is now focused on research, including the impact of nutrition on the aging process and the role of nutrition on immune and inflammatory responses. She's received numerous awards and honors for her work and has held many leadership positions, including her current service as the vice chair on the IAFNS Board of Trustees. So with that, Samin.
Thank you so much, Gwendolyn, and good afternoon, everyone. What I like to, here are my disclosures, and I also serve on advisory boards for some companies as well as government, but none of which conflicts with what will be presented here. So here's the outline of my presentation. I would like to start by talking a little bit about the function of the immune cells and how they respond to infection, so that we can all be on the same page. The cells of the immune system are scattered throughout the body, where they can face pathogen in anywhere, and that is why we can see the consequences of activation of the immune cells in different parts of the body, including in the brain. When the cells of the immune system encounter an invader, in this case a pathogen, for example, there are two types of responses that they produce. One is a cell-mediated immune response, which is production of antibodies as well as dysfunction that allows them to kill the pathogens directly. And they also produce a lot of inflammatory products, including cytokines and lipids, that can also help with the process of the function of the cell-mediated immune cells and the ability for them to be able to get rid of that pathogen. We need both of the cell-mediated and the inflammatory responses in order to mount an effective immune response against an invader. If there is hyperreactivity of the cell-mediated immune response, that causes the host to be susceptible to many diseases, including infection, autoimmune disease, cancer, and arthritis. On the other hand, as was mentioned before by Chris, if you have uncontrolled chronic inflammatory response or overactive inflammatory response, that can cause a sort of a chronic inflammatory response that can contribute to many of the chronic diseases that you're all familiar with, Alzheimer, neurological disorders, pulmonary diseases, cancer, and diabetes, and cardiovascular disease. Now, what happens with aging to our immune system? There is a lot that actually happens. With aging, there is a dysregulation of the immune system, in that you have a decrease in cell-mediated immunity, particularly of the function of the T cells, and those are the cells that are engaged in fighting against viruses and bacteria. And on the other hand, there is this increase in inflammatory responses, which Chris referred to as inflammaging. So when you have, and this is just showing you one example of heightened inflammatory response with age. This is comparing inflammatory products produced from young and old macrophages. And you can see that many of the inflammatory cytokines, there are much higher in old compared to young macrophages. And you also have some of the inflammatory lipids, for example, prostaglandin E2, that is much higher in the old compared to the young. And you actually see this in other tissues of mice, as well as in other species, including in humans. Now, the reason this is important is that there are the age-associated change in inflammatory markers predisposes them to many of the diseases that we see increase with age, which Sarah referred to: heart disease, diabetes, cancer, and so forth. On the other hand, as I mentioned, there are a lot of changes that happens in the T-cell mediated function, including their increased ability to proliferate in a way that they become senescent. It's a very good example of senescent cell. Immune cells are a very good example of senescent cells that Chris referred to, because they can't proliferate, but on the other hand, they are very inflammatory. And this can cause, is sort of reflected in the whole body by inability to produce effective response to vaccine and ability to kill the bacteria and viruses that we encounter effectively. And overall, then it causes increased susceptibility in older people to infectious diseases, as has been really sort of highlighted by the current epidemic of COVID-19 and also with cancer. So one of the things that the diseases that the older you are particularly susceptible to are infectious diseases, including respiratory infections and as well as gastrointestinal infection, urinary tract infection, and as I mentioned, COVID-19, which is also a respiratory infection. And there is a huge cost associated with this. The number that you see here is actually old. It is not much higher and doesn't even include the cost associated with COVID-19. So now, how can we interfere with this dysregulation of the immune response, which seems to be so important in terms of the health span of the older adult? And our focus has been on nutritional intervention, and there is a good reason for that. There has been for many years, it has become clear that if you have deficiency of any of nutrients, or insufficiency of any of the nutrients, you can impair cell-mediated immune response and also increase uncontrolled inflammation, which then predisposes the older individual or the undernourished individual to infection. But what is also interesting is that the infection itself can compromise the nutritional status of the host. This is an example from lung of animals who have been exposed to influenza virus, for example, and you're looking at the level of tocopherol, vitamin C, and glutathione after the animals have been exposed to influenza virus, and you can see a steady decline in all of the three antioxidant nutrients. This is also seen with other viruses, for example, in this case, bovine diarrheal virus, which is a member of coronavirus family. And again, you can see a decline in the level of vitamin E. And this is also true for what happens with the level of vitamin D in serum. There is a decline in vitamin D level as well, indicating that perhaps these nutrients will be important in terms of how we would deal with coronavirus. Now, as Sarah mentioned, a significant portion of the global older population have nutritional problems that are exhibited as both undernutrition as well as which is referred to particularly in terms of macronutrient deficiencies, as well as with overnutrition, which is manifested in terms of obesity. So these are some of the nutrients that have been shown to be compromised with aging. Several of the micronutrients that I'm sure many of you are familiar with. What is also interesting is that there's a good percentage of the older adults that are overweight and obese. And this is, we know obesity is a global problem, but it does manifest itself in the older adult as well. And what is interesting is that obesity itself has been shown to be associated with deficiency of some of the micronutrients. So it becomes sort of a vicious cycle when you are older as well as overweight and obese. And again, both undernutrition, obesity, and aging, all of them can impact the ability of the immune cells to respond to infection, and also how we would control the inflammatory response that comes pursuant to exposure to a pathogen. And if you're older and you have a particular deficiency of the nutrients and you're also overweight, then you can see how this becomes even a bigger problem. Now, how does nutrition affect infection? There are many ways that nutrition can impact the ability of body to respond to infection. One is that it can change the risk for acquiring infection, including interfering with pathogen entry, regulating cell-mediated immune response, and also vaccine efficacy. It can also change morbidity from infection by regulating oxidative stress and inflammation, and it can reduce pathogen virulence and through its impact on the microbiome. Now, I just want to give you, there are many, because of all of this, there's been a lot of interest in looking at the impact of nutrition on the immune response and inflammatory response of the older adults. And there are many examples of them, and what I present to you here does not mean to be comprehensive. I've just selected a couple of micronutrients as well as a whole food, and then a mixture of fruits and vegetables to give you examples of what can be done in terms of intervening with the changes that occurs with aging in immune and inflammatory responses. One concept that I want to bring about that is important to keep in mind is that when it comes to the relationship between nutrition and immune function, it's not a direct line. In other words, more is not always the better. It is pretty much more of a curvilinear type of relationship where, if there's deficiency, obviously you impair the immune response. But as you increase the level of nutrient, there is an optimal level of nutrient that results in optimal immune function. And then after that, you don't see any benefit, or there might actually be some adverse effect. Now, this optimal level might change if you're older or you're under a stress or there's some other have some other diseases, but there is always an optimal level, and I think that's an important concept to keep in mind. So one example that I wanted to share some data about with is vitamin E, which is found in fats and oils and in things like almond. And we have shown, as well as others, through both preclinical and clinical studies, that increasing the level of vitamin E beyond what is currently recommended can improve the immune response in healthy older adults. And I just want to show you a couple of data points from some of these studies. This is a study in which we supplemented healthy older adults with either a placebo or 60 or 200 or 800 international units of vitamin E for about 4.5 months. And then we looked at the response of the older adults to antibodies. And one antibody that we looked at was hepatitis B, vaccine actually, hepatitis B. And as you can see, as we increased the level of vitamin E, we got better response to hepatitis B vaccine, but 200 actually was the optimal level. We didn't see further benefit by increasing the level to 800. Similarly, another measure of immune response, which is referred to as delayed type hypersensitivity skin response, was improved in subjects that were supplemented with vitamin E, and we determined that the 200 international unit was the optimal level for the healthy older population that we looked at. This is from the work of Van Tis et al., where they looked at impact of supplementation with vitamin E on inflammatory cytokines in serum. And as you can see, both in normal lipidemic and hyperlipidemic subjects, those who received vitamin E had significantly lower level of the inflammatory cytokine. So vitamin E seems to have the ability to regulate the immune response in terms of improving cell-mediated immunity and reducing inflammation. We looked at this in context of influenza infection. We used a model of influenza infection in animals. It's a mice model, which very much replicates what happens in humans, where we expose mice, young and old, to influenza virus, and the mice were either fed adequate level of vitamin E or 500 parts per million for a month, and then we looked at how these mice responded to infection. You can see that these mice had significantly less viral titer in their lung, and as you can see, the mouse that were given adequate vitamin E level had a very heightened inflammatory response to the virus, as you can see in the middle and the right-hand side panels, but those who were given vitamin E had a much muted inflammatory response to the virus. So it seems to control the inflammation. And this was reflected in the clinical outcome of the study. One of the things that happens with influenza infection is that there is loss of appetite, and if it continues, then there is loss of weight. And you can see that the animals who were supplemented with vitamin E had significantly less weight loss compared to those who were given adequate level of vitamin E, and also they were eating a lot more than those who were not supplemented. We then wanted to see whether there was an impact in terms of infection in humans. So we used what we had found to be the optimal level, the 200 IU per day, and supplemented nursing home residents for a year with a placebo or with 200 IU per day of vitamin E, and documented their respiratory infections. You can see that those who received vitamin E had significantly lower risk for respiratory infections, particularly of common cold. Now, another nutrients that I want to talk a little bit about is zinc, which is found mostly in animal products. We have found that a significant portion of older Americans have low serum zinc level, and which we consider being less than 70 milligram per deciliter. About 30% of nursing home residents had low serum zinc level, and 22% of independently living elderly had low serum zinc level. What was interesting was that the elderly who had low serum zinc level had significantly higher incidence of pneumonia, duration of pneumonia, as well as higher antibiotic use and duration of antibiotic use compared to those who did not have a low serum zinc level. So zinc seems to be important in terms of the of susceptibility to pneumonia. But this is an observational study, and what we really need to do is to test this in a double-blind placebo control to see if, in fact, if we supplement older adults that are low in serum zinc, would we reduce their incidence of pneumonia? We have some evidence for that from the work that was done by Dr. Parsad, where he supplemented older adults in a smaller study for a year with zinc, and then looked at infection and upper respiratory infection. He did see a significant reduction in overall infection, but didn't achieve a statistical significance in respiratory infection. But it was a small study, and the zinc status of the subjects were not known. In order for us to do a clinical trial, we did a smaller study to see whether supplementation with 30 milligram per day of zinc would correct zinc deficiency in older adults who are zinc deficient, and what the impact of that would be on immune response. And what we found was that we can correct zinc deficiency with 30 milligram in majority, but not all of the older adults who are zinc deficient, and that is associated on average with improvement in T cell function. So we've been fortunate to receive funding to now take this into a clinical trial, but first we want to actually determine the optimal dose and see whether a higher dose of zinc will be more effective than 30 milligram per day. Zinc can also be important in terms of interfering with pathogen entry, particularly in terms of coronavirus, as some in vitro studies have indicated. Now, what I like to switch to is to look at what would be the one of the ways that we can look at the impact of nutrition on immune responses, to look at individual foods. And in this case, we looked at wolfberry or goji berry, which is very much consumed in the Asian countries, and it is presumed to have beneficial effect in terms of immune system. So we wanted to know whether, in fact, goji berry could improve vaccine efficacy. And so we used an animal model to replicate what would happen in humans, in which we supplemented the mice with either with wolfberry or without wolfberry, and then we vaccinated them. And then after vaccination, we challenged them with the actual influenza virus and looked at the outcomes. What we found was that this is looking at the viral titer to influenza. You can see that vaccine is obviously the antibody. You're looking at the left-hand side at the antibody titer, and you can see that when you give the vaccine, obviously there's higher antibody titer. If you give the vaccine with wolfberry, there's significantly more antibody titer to influenza, but wolfberry alone does not change the antibody titer. And this is another way of looking at the antibody titer. This is associated with better clinical outcome. You can see that animals who were given the wolfberry after vaccination and after when they were exposed to influenza virus, they did not lose as much weight as those who were not supplemented with the wolfberry. And similarly, we noticed that the animals who were given the wolfberry had much less mortality from influenza compared to those who were not given the wolfberry. Obviously, vaccination was effective, but the combination of vaccination with wolfberry was much more effective. And with that, I want to summarize, and I hope that I've been able to indicate to you the importance of the changes that occurs with aging in immune and inflammatory responses and its implications for diseases that are associated with aging. And while we need to learn a lot more, it is encouraging that there are at least interventions, nutritional interventions that could be used to try to mitigate these changes in immune and inflammatory responses with aging and change the trajectory of the diseases that would be associated with aging. Thank you so much for your attention.
A number of questions that have come in that we can cover in the next 20 minutes or so. One of the first questions that came in that I'll go ahead and start with is, and I did not know this, today is World Obesity Day, right? And as we think about it, then I'll frame this up as kind of a larger overarching question. You know, when we talk about chronic disease, obesity isn't always mentioned. And, you know, just, I guess, real quick comments perhaps from each of you around obesity versus obesity and whether it should be considered a chronic disease. And then we'll dial into some other questions.
If you would like me to start.
Yep, absolutely. Why don't you start on this one, Sarah, please?
So I'm dodging the question a little bit, and I hope you indulge me. One of the biggest challenges as we get older is our calorie needs go down. And as a result, it becomes more challenging to acquire the proper nutrition with a healthy weight. And I think that the person who asked the question is targeting one of the biggest challenges we're facing with an aging population: how to achieve optimal nutrition while being able to manage weight. And I will hand the podium over to my colleagues to answer and make more comments on obesity. Thank you.
I mean, if you like, I'm happy to jump in here. So I think it's hard to separate obesity from chronic diseases, and I think the question is whether it is a chronic disease by itself, or is it the diseases that are associated with obesity. So that's, I think, difficult, but I don't think that we can talk about chronic diseases without talking about obesity. And certainly in terms of the aging and an immune response, there are so many similarities between obesity, what happens with obesity to the immune system, and what happens with aging, so that, you know, I've been sort of tossing out the referring to obesity as sort of a accelerated aging, because you see the decline in the cell-mediated immunity, the increase in inflammation, more susceptibility to infection with obesity, and then you have all the chronic diseases like, you know, diabetes, like cardiovascular disease. So it is very difficult to dissociate, and I don't know whether we can call obesity by itself a chronic disease. It's certainly a big risk factor for chronic diseases and infectious diseases in this case.
Great, thanks.
I would echo both of those. I would also say that it's almost a kind of a feed-forward loop, if you will, where obesity can drive chronic disease, which then can change the way our bodies respond, which can drive obesity further. It's kind of a feed-forward cycle, and we certainly see this with age, but also obesity can drive other age-related diseases the same way as well. So it's, as both of my colleagues said, it's very hard to tweeze them out from each other.
Great, thanks. So with that, Dr. Wiley, got two big questions, and they both have come up a couple of times, okay? So I'm gonna ask the first one first, which is, of the mouse studies that you showed, they were maintained under specific pathogen-free conditions, right? So that means that you don't have to worry about microbial pathogens and stuff like that. So the question then is, when we go to humans, right, and treating humans with senolytics, do you have to worry about increased susceptibility to infections?
That's actually a really fun question. And it's a really complicated one that I could probably give a half-hour answer to that you wouldn't want me to do. But it is really an important one. So let me do some really broad, very quick hits on this. One example of this is actually that from the caloric restriction studies, we know that once you actually start introducing pathogens, calorically restricted animals are actually more sensitive, not less, to many, many pathogens. So that is a problem right there. But more broadly with senescence, it kind of depends. There are studies out of the Niederhofer lab at the University of Minnesota showing that actually eliminating senescent cells actually allows aged animals to survive better in response to challenges from, for example, murine coronavirus. Those aren't published yet, so I don't want to really do much more than, give her a signal boost at this point, because obviously they haven't gone through peer review yet. But there is some evidence to go both ways on it, and it is a little tricky always to translate to people who are, of course, going out and getting exposed to all sorts of things. But also, we're also more genetically diverse than most model mice as well, which does play a role as well, in that our genetic background may have more influences. So some of us may be more susceptible or less susceptible depending on what our particular genetic background is.
Great, thanks. And then other question for you, Dr. Wiley, that I'll ask is, how much caloric restriction is needed to see the effect, right? Probably have heard that one a couple times, but the results are pretty compelling, so folks are wanting to know more.
So I'm probably not the best person to answer this at the HNRCA. There are actually people that work on caloric restriction that know a lot more than me that'll be coming up in future talks. Actually, maybe Sarah and Samin would actually know this more than me.
So I can actually say, at least the studies that we did, this was a multi-center study, which I was an investigator in, and the study at Tufts was PI'd by Susan Roberts. But we looked at the impact of calorie restriction in humans, for calorie restriction for 2 years in humans, and we looked at 10% and 20%. My focus was obviously on immune and inflammatory responses, and both with actually 10 and 20% calorie restriction, we did see some beneficial effect in terms of improving T cell function and reducing inflammation. So you don't have to go to 60%, 40% calorie restriction to see the benefit. And this was, of course, in overweight subjects that were not obese. And I would think that if you're not overweight, you might not even need to go to that level of calorie restriction. And we really don't know if you're a person of a normal weight, would calorie restriction be beneficial to you or not?
That's great. So perfect transition, right? Because I was going to ask the next question to Dr. Madani. And this is in a different perspective area, and it has to do with dietary guidelines. And, just what are your reflections on whether the current level of nutrients recommended by the dietary guidelines is adequate or optimal for a healthy immune response?
So what I would say is some of it is most of it is a speculation, but we do have some data that would support what I would say. I think, unfortunately, the immune system has not been looked at in the context of requirement, which has been sort of one of the things that I've been trying to get across, that we need to look at the immune system if we're setting recommendations, because it's a perfect way of determining, it's a perfect system. It's accessible, it's biologically meaningful, and it's responsive to nutrition. So why not use it to determine the requirements for nutrients? So having said that, for the most part, we haven't looked at it. So I don't know whether the levels are adequate or not. We do know that, for example, for older population, when we did do a dose response study with vitamin E, that the level that we found was much higher than what's being currently recommended to, produce the optimal immune response. So I think we need to do more of those studies to determine what the optimal level of nutrients is for the immune function, and then perhaps that would impact how we would make recommendations in terms of the nutrients. But unfortunately, we really haven't looked at it.
Great, thanks. Okay, so another question coming in here is whether there are the same beneficial effects with calorie restrictions independent of protein intake, or do you need to reduce the intake of protein as well? Yeah, go ahead, please.
So I think the studies that have looked at calorie restriction have really just restricted calories and not the amount of proteins and other micronutrients. So it is the benefit that it's seen is because of restriction of the calories and not reducing proteins or other micronutrients.
Okay, okay. So inflammation.
I do know, actually, sorry,
Chris, please.
I actually do know people who are working with specific macromolecule restrictions and whatnot. I guess that's a signal boost to some of my colleagues at Buck, like Pankaj Kapahi. But it's, yeah, there's definite ways where you can tweak individual macromolecules and get similar effects. CR is very broad, right? Whereas changing carbohydrate consumption versus protein versus lipid, those can have very different effects. So it's a little bit complicated, a little bit difficult to tweeze out. I would really recommend people go and kind of dig through literature more than I have on the subject, but I definitely know people are working on it.
So just to clarify, I didn't mean that changing the other macronutrients wouldn't impact the immune function. In fact, we've done a lot of studies ourselves looking at different types of fats and in collaboration with our colleagues at the NEPS lab, with looking at protein level and so forth. But what I meant in terms of calorie restriction, particularly, they had looked at restriction of calories in those studies and kept the other macronutrients and micronutrients impact. But yes, certainly, if you change the level of the fat in the diet, the type of carbohydrate in the diet, and the protein, you're going to see an impact on the immune system.
Gwendolyn, can I just jump in to add to this great conversation? I do believe this will come up in the future webinars, but when we talk about manipulating, for example, calories, protein, and individual nutrients, we are presenting it for purpose of simplicity as one size fits all, and that every individual responds the same way. And I just want to clarify for your audience that there is growing recognition in the nutrition field as well as the medical field that we do respond very differently to the same foods, the same nutrients. And there is a huge initiative right now through the National Institutes of Health about what they're calling nutrition for precision health. So these are incredibly important questions. And I would say right now that we're just starting to recognize that there is huge inter-individual variability. And I truly believe that the HNRCA scientists will make a major contribution to understanding why some people really should follow a calorie-restricted diet. I hope I am not one of them. And other people are going to benefit more from a protein-restricted diet. So I just wanted to put that out there for your audience.
Great, thank you. We've got a question here, Dr. Wiley, and this actually gets back into my research, so I get to ask it because I'm looking at it, which is thoughts on telomere length as it relates to biomarkers of age.
Oh man, that's a big question. Certainly we know that short telomeres can drive cellular senescence. That's one way we get senescent cells. Whether it is a major driver is a bigger question. We know that mice age, but we also know that they have active telomerase that maintains their telomeres. We also know that most inbred mouse strains have telomeres that are kilobases longer than human telomeres, and yet mice still get old, and they get old a lot faster than us. So there seems to be a role for telomeres. What it is, it's not as simple as telomere length for certain. So that creates a lot of other questions about this, and it's you know, this is the kind of thing where I refer to people like Elizabeth Blackburn over myself if you want to find out a lot more about it.
Okay, great. Just thought I'd bring that up. So I'll just kind of pull in a question here, perhaps for all of you, which has to do a little bit, Dr. Booth, with what you were talking about, NIH's new research focus on precision nutrition. You know, are we at a point of wanting to look at providing, I'll say, recommendations based on at the individual personalized nutrition level versus population subsets and all of that?
Well, I can start, but I'm going to rely very heavily on my colleagues for adding their opinion. Are we there yet? Absolutely not. I think we're still grappling with some of the more general concepts, and certainly with the context of older adults, particularly when you're getting to the population over 85. Somebody who is 85 is going to have very different dietary requirements and needs than somebody who's 65, yet they're all lumped together. But I do believe that there is sufficient data out there in terms of the gene-nutrient interactions, for example, nutrient-drug interactions, where we do have some basis for moving forward in terms of the research. But do I believe the recommendations are there? No, I think it's very premature at this point. Samin, Chris, thoughts, please.
I mean, I totally agree with you. I think we're not there in terms of making individual recommendations, although I think, as Sarah said, we know that there are variations. I can give you one example. For example, when we were looking at vitamin E and pneumonia, it became clear that the impact of vitamin E, whether it would be protective for pneumonia or not, depended on what genotype the subjects had. And so certain genotypes, vitamin E was protective, in others it was not, which, you know, makes it now understandable why when we do these clinical trials with nutrients, sometimes we find results that are totally unexpected. So I think it is very important to consider the individual variations, but we're not at a point where we can make that kind of recommendations. To be honest, I don't think that we are there to be making really good recommendations. I mean, for the most part, we have. I don't want to discount all the good work that has been done, but we are a long way still to come up with general recommendations. There is still work to be done there, you know, for whether you have diseases or not, whether you have, you know, what age you are, and all of that stuff. But it's all very exciting. I think it provides opportunity to do a whole lot of research.
You know, one of the themes you kind of keep coming to when you work in aging research is variation. The older you get and the longer you go, the more variability you get. It's so difficult even to study completely inbred mice because they become hyper variable the older they get. So it's just very difficult to know exactly how things work. We know that in the general population that lots of diseases come up with age, but there are people that don't age the same way, right? That they stay healthy much longer. We talk about centenarians, for example, but we also just see massive variation from twins as they get older, right? Because they're genetically identical, They could even live very similar lives, and nevertheless, as they get older, they develop diseases at different rates too. And that's so we can completely even take genetics out of the equation and still see increased variability with age. And that's a really, really hard thing to tweeze out.
Excellent. Great. Go ahead, please.
I was going to say, I wish I had a slide there that I took out to stay in time, but it was showing with age in humans the immune response, and you could see 90 years old that had an immune response that was like a 40 years old, and at 30-year-old that had immune response that was like a 60-year-old. So it's very valuable. So obviously genetics, and I mean, Sarah had a very nice slide that kind of looked at all this variables that influence how you respond and how you age, which had the genetic and the environment and diseases and all of that. So I think not to say that sometimes when you point out all these complexities and heterogeneities, people get disappointed and they discouraged and say, well, we don't know anything. It's not really true. We know a lot, but I think it is to be conscientious of the factors that can affect, and to question when you hear something or when you look at some result and a result from publications, to sort of understand and put it in context.
Absolutely. Completely agree. So I know we are now at the end of our time. Let me start by asking folks and reminding folks to join us in 3 more weeks for the next webinar in this series. This next one's going to be focusing on balancing healthy weight while meeting nutritional needs for bone and muscle health. With that, also want to note, and I'm sure this will come up in the rest of the webinars that we have, that there's so much exciting science going on in this area, whether it's exploring gene interactions, whether it's exploring different components from a distilled or combined perspective, either which way. It's all very, very exciting. So with that, I will note as I go to the next slide, which has my contact information on it. Want to thank all of the attendees for taking their time out today to join. We did not get to all the questions, unfortunately, but know that we will be working to have this webinar available for viewing later on this month, okay, so that you can rewatch it, as well as share it perhaps with other colleagues who could not make it. Also want to really extend a sincere thank you to our presenters today, Dr. Booth, Dr. Wiley, and Dr. Madani. I think this was great and informative, and I know everybody learned a lot. So with that, thank you, everybody, and look forward to seeing you again in a few weeks. Thanks. Bye-bye.
Thank you.
Bye.